So in this video, I'll be telling you a real-life story while simultaneously explaining the concept of DNA profiling. So there'll be two parts, so make sure to watch both to understand everything about DNA profiling that you need to know, specifically for the IB. Now, so let's get started. So pretend you're a cop or you're a scientist. Pretend this is your job, okay? Kind of like, I don't know if you guys know, if you know the series Bones, where the scientist and the cops work together to determine who the murderer is of a specific crime scene. Now pretend you're one of those people and you're in this room. Based on initial impression, you can see this was a crime, right? You can see some blood. You can see the house is completely messed up. There's a murder weapon perhaps. Maybe this is like a witness. There's a photographer. Okay, pretend you're in this situation. What do you do first? Like if you're the cop or the scientist, what do you do? So normally the first thing we encounter is the witness, right? So pretend this is the victim. So normally the first thing we see is the victim, right? Because normally they're lying dead on the floor or something like that. So pretend the victim's name was Georgie. Obviously you can see Georgie isn't here anymore. He was removed from the crime scene because that's normally what they do. Now, so you normally as a cop or a scientist want to make notes. So normally the notes are, you want to be able to find out who the victim is and who's a murderer. But in this case, we already know who the victim is because we saw it's Georgie. In this case, it wasn't so complicated because the victim wasn't difficult to find. He was right there on the floor. But the tricky one in this case will be, who is the murderer? Obviously, the murderer isn't on the floor either. He's gone, right? You can see the footsteps. He actually left the house. So this is what we want to find, and that's the purpose of DNA profiling. The purpose of DNA profiling is to be able to match the DNA from a sample. So in this case, the sample is the blood from the crime scene to the specific suspect or the murderer. Okay, so that's all DNA profiling is. So in order to do DNA profiling, you need to carry out some steps, and that's what we're going to go over in the next two videos. Now, the hard part for you guys, for everybody, is to find out specifically who the murderer is, right? So the question is how? How do we do this? Let's see if you guys know some ideas. So what would you do in this case? So we know there was a crime scene took place. What do you do first? So normally, first what they do, simultaneously, they will try and find potential suspects, right? So in order to do that, they normally ask a witness or something, or like a parent, if there's any enemies of the victim. Did he have a friend that was angry at him? Did he have a did he have a neighbor that was just that he had problems with, and all these things? So in this case, that's the first thing they did. So these cops went to interview this witness and asked for potential suspects, and that's what the witness gave them. So in this case, we have a bunch of suspects. Maybe you know all of these three. So we have Ronaldo, we have John Wick, and we have Johnny Bravo. Okay, all of these three were apparently suspects, and we can all see why John Wick was, because he's a murderer and obviously he's killed a lot of people. And then Johnny Bravo, apparently he was just a neighbor that was kind of like a stalker, so he was a potential suspect too. And Ronaldo was apparently just his best friend, but you know, things can always go wrong with best friends. So in this case, these are the three suspects. So we have the victim, we have the suspects. And now we're going to do DNA profiling to find out which one of these three it is. Do you guys know which one of these three it is? Can you have a guess? Now, let's actually find out. So the second thing we do, you do this, the scientists kind of do this. They will specifically focus on the blood because that's all they have. So the scientists believe that this wasn't an easy murder. As the murder was happening, the victim managed to spill blood of the murderer. So this is not blood from just the victim. This is blood from actually both. So we know you can also take fingerprints, right? That's also another option. But the murderer was smart in this case. So he actually wore gloves. So everything he touched, there's no fingerprints on it. So they have to rely on the blood. So let's see how they do it. So this you don't have to know so well, but just know that blood has a lot of cells in it, right? Just like the rest of our body. You guys know red blood cells. That's what makes the blood have the reddish color. But the problem is, red blood cells doesn't have DNA in them. But there are other cells inside blood that does have DNA. More the white blood cells. So these are all white blood cells, and they have some DNA in them. Why do I care about DNA? So the reason why I actually care about DNA, or why the scientist would care about DNA, is because DNA is unique to every person. So the DNA that you have is different from the DNA that I have. And so it's kind of like an instruction manual. Every person has a different instruction manual. Now, so this DNA is going to be extremely useful in DNA profiling, because think about it. All they have to do to find the suspect is go interview the suspect, take a blood sample with permission, and then compare the DNA from each suspect's blood sample to the DNA found at the crime scene. And whichever one's DNA is going to match the DNA of the sample, that is likely the guilty one, right? That makes sense, right? So obviously, this DNA profiling does not only have to be used in a crime scene situation. You can also do it with paternity. For example, if a lot of the times there's situations where they're not sure who the parent is. Like, for example, if there's a lot of affairs going on and suddenly the wife has a kid, after a while and they're not sure which one was the husband, they can also use this technique to compare the DNA of the child to all the potential fathers, and the DNA will be very similar between the child and the father, right? So that's another way where it can be used. But for now, we're just focusing on a crime scene, right? Okay, so now you ask me how. How the hell do we do this? So we know now we're going to use this DNA and then try and compare it to the suspect's DNA. But how does this work? Okay, so let's move on to that. That's the whole topic. So normally the problem is at a crime scene is that the DNA is not enough, right? So there will be a little bit of blood on the floor, but normally that's not enough to actually do the testing. So it won't be enough. It's kind of like, think about this. It's kind of like a printer. If you put a little bit of ink into a printer, not enough ink, just a little bit, normally the page that you print will be kind of blurry. It won't be as good as it should be, right? So that's kind of similar in this situation. If there's not enough DNA to do the testing with, then the testing won't be accurate. And that's really risky because you don't want to sentence the wrong person into prison, right? So they got to make sure they have enough DNA to do this testing with. So how do they do that? That's the focus of part 1. So that's the focus of this video. We're going to focus on how they make a lot of DNA from a little bit of DNA. So let me show you. So first what they do is they extract the DNA from the cell. They will literally take the DNA from the cell because they don't need the cell. All they need is the DNA, because we know the DNA is the only thing unique to you, right? And then what they want to do, so this is called extraction, but you don't have to worry about that. You have to care about what they do after they extracted the DNA. So what they do after they extracted DNA is they want to multiply it. Make many, right? Eventually, until they have enough. Okay, so let's pretend they made now many. What's this process called? You can think it's DNA replication, right? Because you're basically replicating DNA. Yes, that's basically what you're doing. You're replicating the DNA to make many. But you're not doing it the normal way. When a cell does it by itself, when a cell does DNA replication by itself, then it's called DNA replication. When you're forcing it, when a scientist is replicating the DNA through a machine, then it's called something else. Then it's called PCR. Maybe you've heard this word, but it's important, so we're going to go into it. So when a scientist makes a lot of DNA from a little bit of DNA, that's called PCR. And the official definition is, it's right there, but what it basically is making a lot of DNA from a tiny bit of DNA. Because think about it, sometimes at crime scenes, you may not even have that much blood. You may actually have just a droplet, and then you need to make a lot of DNA from that, right? So that's the key. So that's all that PCR really is. But you need to know, apparently for the IB, you need to know how this happens, how you go from one to many. So that's what I'm going to teach you right now. I'll go slowly, so just to make sure you guys understand it. But it shouldn't be too bad. So again, the official name of doing this is polymerase chain reaction. This name will make sense. Don't worry. As I do it, I'll explain it. But the beginning at least makes sense. You can think poly. Poly means many. So you're gonna make many DNAs, right? You're gonna replicate the DNA, many. And you'll see why it's called a chain reaction. So let's go. So now they're gonna take this blood, these scientists, from the crime scene that was on the floor, and they're gonna take it to the scientist, and the scientist will go into a lab and do PCR with it. They're gonna put it into a machine. And let me explain what this machine does. So there's 3 steps. It's not difficult. Just try and understand it. So here we got it. So they take this tiny bit of DNA. Nope, it's not working. Okay, so they take this tiny bit of DNA. So here it is. That DNA. You know that DNA is double-stranded, right? You got two strands connected to each other. That's why it forms this like helix shape, right? But don't worry about that for now. Just know you got 2 strands. And right now we only have 1, and we want many of these. So how are we going to do that? So what they do first is they put it in an extremely hot temperature. So they heat it to 95 degrees Celsius. And what this does is it actually breaks the bonds in between these 2 strands. So what it's going to do is it's going to separate these strands into one up here and then one down here. It's going to separate them. And that makes sense, right? Because think about it. If you put yourself in 95 degrees Celsius, you're probably going to split apart too, right? So this part is logical. They heat it to break the bonds in between the 2 strands, and that will separate them. This is very similar to DNA replication. Remember, in DNA replication that the cell does by itself, instead of having temperature to break these 2 strands apart, there's an enzyme, right? And this enzyme is going to cut these hydrogen bonds. But now, this is not normal, so we're doing it by machine. So temperature is going to do that for us. Now, here comes the trickier part. If you can understand this, you got the whole process. So think about it. Right now it's really hot, but now they've been separated. So now what they're going to do is they're going to wait for it to cool down. So they're going to wait for the temperature to drop a little bit. And you might ask why? Why do they want to do that? So they want to do that because if the temperature stays high, then they can't add any DNA onto these strands, because as soon as they add them, the temperature will just cause them to break again. So you won't be able to actually form more DNA. So that's super important for to wait for this it to cool down. Now, as soon as it's cooled down, they're gonna add something called a primer. You need to know what this is. So a primer is basically a thing like this. This is dark green spot. It's DNA that is added, and it will serve as the initial spot where DNA will be synthesized from. So it's kind of like the start button. Okay, so that's what happens. The machine will add these primers that will bind, and that will cause the rest of the DNA to be synthesized from that primer. You can think primary means beginning. So you can think it's the beginning DNA. Don't worry too much about the science behind it. You just got to remember this. So primary, so it's the first part that's being added. And obviously it happens to both. So it's going to happen to this strand here and this strand here. Now, as soon as it's added, now what's going to happen is this. Ignore this for now. Just focus on this. So now what's going to happen is once the primer's been added, free nucleotides will be added. So these things right here are free nucleotides. And basically, you know how the things holding 2 DNA strands together are nucleotides, right? It's the A for adenine, the T for thymine, the C for cytosine, and the G for guanine, right? A will bind to T, and G will bind to C, right? Now that's the same thing here. So these are basically a bunch of letters floating around. Now, it's like a puzzle. So all we want to do now is put these nucleotides in the right places until the entire thing is synthesized, right? So what's going to happen is this DNA polymerase, or Taq DNA polymerase, so that's this thing right here. This thing is an enzyme. Any word that ends in ASE is an enzyme. And we know enzymes kind of catalyze reactions. And that's what it's going to do. It's going to make sure it's going to put all these pieces in the right places. So essentially, it will basically like move and bind to this free nucleotide and then add it to the correct spot. So it's going to go add it, and then it's going to move and go add the next one. And then it's going to move and add the next one until the entire thing is synthesized. Because right now, we managed to synthesize 2 copies, this one and this one, from only 1 copy. So already we have doubled the amount of copies, right? Because we can think this strand right here is going to be identical to this strand right here. And then this strand right here that has been newly synthesized will be the exact same as this strand here, right? So we actually have the exact same DNA as over here. Now over here, this bottom strand will be the exact same as the bottom strand here. And the newly synthesized strand will be the exact same as this strand here. We managed to make 2 new copies of DNA. We replicated it, all right? So this is the first cycle. After 1 cycle, we managed to make 2 new DNA copies from 1. Now, if we continue on, what do you think will happen? Now think about it. Now we're going to repeat the exact same process, but with 2 DNAs, right? So what will happen? In the second cycle, we'll end up making 2 more, right? 1 and 1 from this DNA, and then 2 more from this DNA. So already we have 4, right? So what's going to happen in the third cycle? How many do you think we'll have now? After the third cycle, we'll have 8, because each one will make 2 more. Each one will make 2 more, right? So fourth cycle will be even more. And then eventually, after you do it like 30 times, you're going to have billions or millions, right? So after many, many cycles, you're going to have so many. So you can see how this kind of process goes exponential. It goes faster and faster and faster than what you're making. Because each time, it's essentially doubling, right? So in order to know, a very good way to remember how much DNA is made every cycle, you can use this equation, 2 to the power of n. So for example, if I ask you, how much DNA will be made after 2 cycles? Then all you have to do is plug the number 2 instead of n. Okay, so n stands for the amount of cycles. So that will be 2 to the power of 2, 4. That's true, right? So how about if the teacher were to ask you, how much DNA would you have after the 3rd cycle? Then you'll just plug 3 in. That will give you 8. So now we have 8 total DNA copies. And how about, and if she's like 30, then all you have to do is 2 to the power of 30, right? So you can see how quickly you made millions or billions of copies of DNA. Now that's the whole point. So that's the first step in DNA profiling. We first want to make enough DNA. So after you've made billions of copies of DNA, now the scientists are ready to go actually go and test to compare if the suspects, which suspect matches the DNA of the crime scene. And that way we will know the murderer after all. So the next video will actually explain how they use this many copies of DNA that was made by PCR, so made by polymerase chain reaction from the DNA at the crime scene, and how they use these copies in gel electrophoresis. So this is a process that I teach in the next video. So I'm gonna explain how they use this millions of DNAs in gel electrophoresis to prove who the murderer was. So if you want to know who the murderer was between these 3, gotta check the next video.